Hi, welcome to my YouTube channel Bio Explorer. I am Dimujjaratna. This is an educational channel and this channel will be very useful to improve your biological knowledge. Today in this video, we are going to talk about an essential instrument which is widely used in the field of biology. It is microscope. Microscope. Microscope is an instrument used commonly in school or medical labs to examine objects that are invisible to the naked eye. Advancement of the cytology is mostly based on the microscope. The discovery and early study of cells progressed with the invention of microscope. There are many types of microscopes. They can be grouped according to the method used to produce an observable image. Most popular types of microscope are optical microscope and electron microscope. Optical microscope is able to produce images using visible light refracted by lenses. It is the most common microscope. In electron microscope, an electron beam is passed through the specimen and the pattern of electrons passed through the specimen is used to produce an image on its screen. Parameters in a microscope. Resolution power and magnification are important parameters which can be seen in the microscope. Magnification is the ratio of an object's image size to its actual size. Usually, the maximum magnification of light microscope is 1000 times than the actual size of the specimen. Resolution power is the minimum distance between two points that can be distinguished as separate points. It is a measure of the clarity of the image. Magnification is limited due to the resolution. Resolution power of light microscope is 0.2 micrometers while resolution power of a human eye is about 0.1 micrometers. The compound light microscope. This is the most common type. It is commonly used in school laboratories and it is used in medical laboratories as a diagnostic tool. First, we will look at the parts of a light microscope and their functions. This type of microscope is generally made up of structural parts and optical parts. The structural parts help to hold the microscope, while optical parts are used to enable visualization of the specimen. The main parts of a light microscope are head, nosepiece, objective lenses, stage, condenser, diaphragm, light source or mirror, eyepiece, eyepiece tube, arm, coarse adjustment knob, fine adjustment knob and base. Structural parts. There are three structural parts as head, base and arm. Head. It holds the eyepiece tube. Arm. It connects the head and the base of the microscope. It is used when carrying the microscope from one place to another. It holds the nosepiece, which consists of objective lenses. Base. It keeps the microscope on the substrate and supports the microscope. Optical parts. Eyepiece. It is situated at the top of the microscope where the eye is placed. We look through the eyepiece to see the image. The specimen is enlarged for the second time by the eyepiece. Eyepiece tube. It holds the eyepiece. Objective lenses. The specimen is enlarged first by the objective lenses. They can be mounted as needed. Each lens has its own magnification power. Nosepiece. It holds the objective lenses. It can be rotated to view the specimen with the matching objective lens. Stage. Specimen slides are placed here for viewing. The stage clips are used to hold the slide in the right position. Mirror. It is located at the base. The mirror, which can be adjusted as needed, directs the light rays into the condenser. It can be a plane mirror or a concave mirror. In some microscopes, there is a light source instead of a mirror. Condenser. It is used to collect and direct light from the mirror into the specimen. Condenser is very useful to produce clear and sharp images. Diaphragm. It is situated under the stage and it controls the amount of light reaching the specimen. Adjustment knobs. There are two types of adjustment knobs as fine adjustment knob and coarse adjustment knob. They are used to focus the microscope, that is, those knobs adjust the distance between the lens and the specimen to obtain a clearer image. How to observe through a light microscope. First, prepare a thin layer of the specimen on a glass slide. Cover the specimen with the cover slip. Light should be able to pass through this specimen. A section of specimen as thin as 5 to 10 micrometers can be clearly seen under a light microscope. Place the microscope on the table so that the base of the microscope touches the surface of the table. Place the specimen slide on the stage as the cover slip is on the top. Secure it with clips. Observe through the eyepiece and insert the specimen into the field of view. By looking through the eyepiece, adjust the mirror, enabling the optimal amount of light to reach the specimen to get a clear image. Look at the microscope from side to side and using coarse adjustment knob, set the low power objective lens about 20 mm above the stage. Lift the low power objective lens using coarse adjustment knob until you get a clear reflection. If you want to zoom in on a part of the specimen, move the slide and place it in the center of field of view. If the image is not clear, it can be focused by fine-tuning. At this point, the high power lens is very close to the glass slide, so the rough adjustment knob should never be used to focus the high power lens. Using aperture of the condenser lens, the light can be adjusted accordingly for clear observation of the specimen. The diaphragm controls the amount of light that enters the specimen through condenser. Electron microscope. It is another type of microscope. A limitation has imposed upon the resolution power of the light microscope by the wavelength of light. Resolution power should be reduced to obtain clear images. Resolution power is proportional to the wavelength. So, in order to reduce resolution power, wavelength should be reduced. Due to this, scientists considered the use of other forms of radiations with comparatively shorter wavelengths. As a result, electron microscopes were developed. Basically, electron microscopes have a higher resolution power, meant numerically lower than light microscopes. Resolution power of an electron microscope is about 0.2 nanometers. Here, a beam of accelerated electrons is used to produce image. Wavelength of an electron is about 100,000 times shorter than visible light photons. In electron microscope, a beam of electrons is focused through the specimen or onto its surface. Electron microscopes have revealed many organelles and other subcellular structures that are impossible to resolve with the light microscopes. The living objects can't be observed through this type of microscope because The electrons react with the water particles in the specimen, therefore only non-living specimens can be observed. So, it is essential to keep a vacuum in the electron microscope. Electron microscopes use magnetic lenses to focus electron beams. The image is created using electron patterns that travel through the specimen or that are reflected from the specimen. Therefore, surrounding magnetic fields can affect the flow of electron beam. It is a disadvantage of electron microscopes. The reflection can't be seen directly with the naked eye, as in light microscope. The final image produced is called an electron micrograph produced by specialized digital cameras. It is a black and white image. In theory, the electron microscope should be able to magnify objects up to 10 to 5 times, but in practice it magnifies just over 5 into 10 to 5 times. There are two types of electron microscopes as transmission electron microscope and scanning electron microscope. Transmission electron microscope. It is used to study the internal structure of cells. Here, the electron beam produced by an electron gun is transmitted through the specimen. Extensively thin sections of the specimen, typically about 100 nanometers, are used. They are stained with heavy metals before observation. Those heavy metals attach more to certain cellular structures than the other areas. The emerging electron beam from the specimen is captured and the pattern of electrons is processed to produce the image. More electrons may get displayed in the regions where structures were densely stained. Scanning electron microscope. A scanning electron microscope captures the surface of a sample rather than its interior. Here, a fine beam of electrons is reflected from the surface of the specimen. Extensively prepared specimens are not needed because electrons do not have to travel through the specimen. The specimen is mostly coated with gold prior to observation. Most of the electrons hitting the specimen are scattered while others are absorbed. This type of microscope is commonly used to produce images of the three-dimensional surface shape of the specimen. So that's for today. Hope you enjoy this video and gain a lot of facts about microscopes. Please subscribe this channel and click the bell icon. See you in the next video.